Good evening, everyone. Thank you for joining us. My name is Michelle Holmes, Director of Client Relations for Avexia Diagnostics, and I will be your moderator for tonight's presentation, GI Stool Testing and the Development and Clinical Uses of the GI Map. I am very pleased to introduce our presenter, Dr. David M. Brady. He has 25 years of experience as an integrative medicine practitioner and over 21 years in health sciences academia. He is a licensed naturopathic medical physician in Connecticut and Vermont, a board-certified clinical nutritionist, and completed his initial clinical training as a doctor of chiropractic in 1991. He currently serves as the Vice President for Health Sciences, Director of the Human Nutrition Institute, and Associate Professor of Clinical Sciences at the University of Bridgeport in Connecticut. He maintains a private practice, whole body medicine in Fairfield, Connecticut. Dr. Brady is also an expert consultant to the professional nutraceutical and nutritional supplement and clinical medical laboratory industries, serving as Chief Medical Officer for Designs for Health, Inc. and Diagnostic Solutions Labs, LLC. He is an internationally sought-after presenter on nutritional, functional, and integrative medicine. He has appeared on the speaking panel of some of the largest and most prestigious conferences in the field, including IFM, ACAM, A4M, IHS, AAMP, and many more. Dr. Brady has published a multitude of peer-reviewed scientific papers and textbooks related to chronic pain, autoimmunity, and functional gastroenterology, and is also featured as a contributing author in the medical textbooks Advancing Medicine with Food and Nutrients, Integrative Gastroenterology, and Laboratory Evaluations for Integrative and Functional Medicine. His latest popular book, The Fibro Fix, was published by Rodale and released July of 2016 and coincided with his hosting of the popular online Fibro Fix Summit in June of 2016 with over 30,000 attendees. Before we begin, I would like to encourage all of the participants to send any questions you may have during the webinar. Please type the question into either the chat or question box and send to the organizer. We will respond to these questions at the end of the presentation, allowing for extra time if needed. A common question is where you can find the recording of this webinar. I will send you a link tomorrow, and it will also be available on our website by logging into your account and going to Client Services. If you are not already a client, please go to our website, www.avexiadiagnostics.com, and click on Become a Client to join us. Before I turn you over to Dr. Brady, I also want to further announce the launch of Avexia Nutraceuticals. We are proud to offer the Avexia line of nutraceuticals, which will transform how clinicians manage the health of their patients by providing products and services to promote optimal health. During the latter part of tonight's presentation, Dr. Brady will speak to you about how Avexia Nutraceuticals are an integral part of your course for treatment. Good evening, Dr. Brady.
Hey, thanks, Michelle. I appreciate you having me on again, and thanks to everyone at Avexia for making this possible. I'm really excited tonight to be able to speak on a topic I have a great deal of passion for, which is basically using GI diagnostics within the context of the functional medicine paradigm in dealing with complex chronically ill patients. And we're going to talk specifically about the uses of the GI map test. And we're going to really center on a lot of the emerging information on the human microbiome. And as we all know, the human microbiome is the hottest area of medical research at the moment, probably for the last 5, if not 10 years, along with personalized immunotherapy in various types of cancer therapeutics. Tonight, even though the human microbiome is vast, it covers many organisms over many types of tissues and surfaces and mucous membranes in the body, we're going to really feature the predominant area of the human microbiome, and that is the GI microbiota. So we're going to be looking at a lot of the different organisms and their genes and metabolites that make up the microbial mass running through that hollow tube in the middle of us we call the GI tract. The Human Microbiome Project has really opened the windows, if you will, onto new ways of looking at our health, looking at disease and looking at the intersection between health and disease. And we're going to be able to apply some of what has been learned from the Human Microbiome Project in the way we manage patients. And we're going to look at some of those issues tonight. As I already alluded to, we really have this unique situation where we have an external environment or a hollow tube containing an amazing number of microbes, toxic materials, foreign peptides in the form of food, and all kinds of different stuff running right through the core of our body, right through the middle of it, technically outside of it, but nevertheless running right through it. And in fact, there's many, many more microbes in that gastrointestinal lumen running through the middle of us than there are cells in our body. And their genes outnumber our genome 10 to 1. So it is a really important area. A lot of these microbes, their metabolites, the genes that they are expressing, the metabolites, the LPSs, the various types of chemicals that are emitted from this microbial mass directly affects our physiology, dances with our immune system, and actually helps determine whether we're healthy or whether we're diseased. And if you don't believe this, let's look at one example. This is an interesting study that originally appeared in the preeminent journal Nature, and then is now being talked about in this paper here in The Scientist, another very high-level peer-reviewed journal. And what this research team did is they took an animal model, they took a wild-type mouse, healthy animals with a normal mouse GI microbiota, so all the normal organisms that you'd find in a mouse's gastrointestinal tract, and they introduced one foreign bacteria into the GI tract of these animals, and that bacteria was called Helicobacter hepaticus. It's related to Helicobacter pylori, which we know from ulceration and gastritis and stomach cancer and so forth. Same genus, different species. So Helicobacter hepaticus was introduced into these animals, and what it did is it caused an extreme activation of a specific type of immune cell called Th17 cells, which in turn produce and release highly inflammatory cytokines known as interleukin 17. This causes an inflammatory storm and essentially creates inflammatory bowel disease in these animals, which is a classic autoimmune disorder, similar to ulcerative colitis in a human being, or celiac disease, or Crohn's disease, or any of those variants of an inflammatory bowel disease. So by simply changing one microbe in the GI tract of these animals, they took healthy animals and created essentially a mouse model of an autoimmune disease, inflammatory bowel disease. And then they introduced one more bacterium, in this case B. fragilis, and B. fragilis presents on its surface a polysaccharide A, or a PSA, which is a molecule that's taken up by other immune cells called dendritic cells, and they activate CD4 T cells and regulatory T cells, which in turn release another interleukin called IL-10, which is highly anti-inflammatory and essentially suppresses the action of the original IL-17 that created the inflammatory bowel disease. So essentially, one bacteria introduced produced an inflammatory storm or a cytokine cascade that produced inflammatory bowel disease. The introduction of yet one more bacteria essentially neutralized it and normalized the animals. So they caused autoimmune disease with one bacteria. They resolved autoimmune disease with one more bacteria. So you might say, yes, this is an animal study. What does this have to do with humans? Actually, a lot. It shows the whole premise that even minor strategic changes in the human GI microbiota, or in this case, an animal microbiota, can have profound effects on the host. And in fact, in the medical literature, we see various systemic disorders that we may not necessarily associate with the gut. Some of them we do. But many of them the average person would not associate with the gastrointestinal tract or it being a GI problem. But we know that they're highly associated with aberrations in the GI microbiota, meaning the wrong kind of microbial balance, a situation we call dysbiosis. And often concomitantly, they have a phenomenon known as intestinal hyperpermeability or leaky gut. And you see on this slide, this is a very partial list of these disorders. The list would be much, much longer if we were being all inclusive. But you see a lot of disorders that end in itis, right? A lot of inflammatory conditions, a lot of autoimmune conditions, a lot of conditions that we don't even consider either of those, but yet are associated with aberrations in the gut ecology. And to give you an example of a disorder that's having profound public health ramifications that is not autoimmune specifically, is the autistic spectrum. Now, children on the autistic spectrum have very different GI environments than normal children. And we're able to determine this with real-time quantitative PCR analysis of the microbial mass of the GI environments of children with autism and without autism. And when we do so, it's very, very striking that there is a large representation of various clusters of Clostridia, the organism Clostridia, in the GI tract of autistic spectrum children that is not resident in normal children. Now, you might jump to a conclusion there and say, oh, well, Clostridia must cause autism. And that is not what we're saying here. This is an association, not necessarily causal, but it is clear that even beyond just Clostridia, there's significant differences in the GI environment and what's going on there in autistic children versus normal children. And it is probably one of the many constellation of pieces or perfect storm elements that need to be present to create autism in an individual. And indeed, autistic spectrum children often improve dramatically. Their functional outcomes improve with fecal transplantation or microbial transfer therapy, which basically means imparting fecal material with the microbiota of a healthy host or a healthy individual into the subject. Now, speaking of stool and stool transplants, it's to stool that we have to turn as clinicians to try to ascertain what is going on in the gastrointestinal environment of a patient who may come in either with significant GI complaints or a patient with complaints that we know are strongly related to aberrations in the GI environment, including inflammatory conditions, autoimmune conditions such as rheumatoid arthritis. We're going to look at some autoimmune thyroid conditions. We're going to look at some examples of MS and many, many more. And what's enabled us to be able to look at the GI environment in a whole new way and establish some of these associations or these connections between certain patterns in the GI microbiota and higher predilection of certain diseases has been the molecular revolution. The ability to use PCR amplification of the resident DNA of whatever organisms are there and to actually be able to detect organisms at the level of the genes or the level of the DNA. This has created the ability to look at a lot more of the GI microbiota than we were able to do so previously with culture type of technology, which was the only thing we had for many, many decades before not too long ago. So we're going to look at the GI map, which is a DNA-based stool test that uses quantitative PCR, real-time polymerase chain reaction with quantitative PCR elements to actually identify what is going on in the patient's GI environment and help us solve a lot of chronic complex diseases. Now, some of the advantages of the GI map are that it uses true quantitative PCR molecular methods, meaning it looks for the DNA of these organisms, and it not only can determine which organism is present, but it could quantitate the level of DNA of that organism, which is very important when you're trying to make clinical decisions such as, does someone have a clinically significant infection with a pathogen that needs to be treated, for instance. Molecular methods using DNA analysis also do not have to rely on whether a microbe or a bug will grow in a nutritive media or on a plate for analysis. That's very, very important because previously, when we had to rely on growing out organisms with culturing types of technologies, we were really able to only look at organisms that would grow in an oxygen environment. So we were looking at aerobes, some facultative organisms, but mainly aerobes, and they make up less than 5% of the organisms that live in your gastrointestinal tract. So right away, we were extremely limited into what we were able to actually analyze. But with DNA or molecular methods, we can look at all of the organisms present, aerobes, anaerobes, and facultative organisms. We don't have to worry about microbial growth during transport after the sample is collected, such as in the collection tube in the FedEx truck while it's trying to get to the lab. Because in a molecular sample, unlike a culture sample, it's dropped in formalin. So the minute it comes out of the patient, the sample, it's put in the sample tube, everything is pretty much frozen in time at that point, and then you can look for the genetic signatures or the DNA, because the organisms do not have to be alive, such as they do have to be in culture for obvious reasons. And with the GI map, the quantitative PCR methods that are used are centering on specific DNA elements of these organisms, including the 16S RNA, the 23S RNA, other elements of RNA, SNPs and genes, and virulence factors, or various genetic characteristics of specific organisms. And this allows us to ascertain different types of information about different organisms, which you'll see coming up shortly, help us make clinical decisions. Molecular technology also allows for very rapid turnaround time because you don't have to wait for organisms to grow. You don't have to feed them comparative nutrients or food sources or what's called a comparative sugars test to try to identify them. Now, there are various types of molecular methodologies out there in stool testing, and some of the big differences are qPCR versus next-gen sequencing. Now, these are both DNA or molecular technologies, but they're inherently different from one another. You may have heard of some of the direct-to-consumer testing, like uBiome. This is a very good test for what it's intended to do, but it was never intended to be a clinical test for doctors to make clinical decisions from. It is really what's called a metadata test. It's a big picture snapshot of one's microbiome. It looks for lots of different organisms, but it doesn't quantitate any of them. It actually reports all the organisms as a percentage of the total organisms. Because on this type of test, you're not really testing pathogens, you're not trying to quantitate, you're not trying to make a decision to treat or not. You're using these types of tests, or more researchers are using these types of tests to develop large population signatures of what a microbiota might look like in different areas of the world, in cohorts on different types of dietary approaches, and they can answer large issues such as microbial diversity, microbial abundance in a patient's GI microbiome, but it's not meant to answer the nitty-gritty questions that the clinician needs to know, which for that they need to rely on a quantitative PCR clinical test like a GI map. So when we look briefly at a report for a GI map, and we're going to go through this in more of a clinical context in a moment, but on the first page, what you're seeing first are pathogens. And of course, pathogens are pathogens. They can cause pathology. They can cause serious gastrointestinal distress. They often cause fulminant diarrhea, not always, but often. Many of these organisms you've heard of associated with food poisoning and other types of gastroenteritis. Things like Campylobacter, Clostridia difficile, enterohemorrhagic E. coli, Shigella-like toxin-producing E. coli, Salmonella, Vibrio, Yersinia. These are all nasty bacteria that you don't want in your gastrointestinal tract. But we also look at various parasitic pathogens, including protozoa that are listed here, like Cryptosporidium, Entomoeba histolytica, Giardia. These are all organisms that are not all that uncommon We get exposed to them in rivers, lakes, swimming pools, but also in food as well. And then, of course, viral pathogens, your adeno and noroviruses, most associated with the gastrointestinal viral flu. And then we can look at situational pathogens, organisms that could be pathogens, but could be benign residents, such as Helicobacter pylori. And we can look at various characteristics of the H. pylori called virulence factors to determine not only is there more H. pylori present in a patient than would be considered statistically normal, but is it likely to cause problems? Is it likely to cause gastritis or duodenitis or ulceration or even gastric cancer? Going further on in the test, we can use the same technology to look at what are called normal or commensal bacterial flora. Some people would call these probiotics, right? The good guys. And some of the ones most known on this list, of course, are lactobacillus, bifidobacteria, and other organisms that do beneficial things for us, such as help us digest our food, help us produce various nutrients, help us keep pathogens at bay, and help express our mucosal immunology in a healthful way. We are going to look at some issues of a phyla level look at the microbiota, looking at the Bacteroidetes and Firmicutes phyla of organisms, and I'll save that for just a little bit later. But then the test will look at opportunistic organisms. These are not pathogens per se, but they're also not beneficial organisms. They're everything in between. And when these organisms overgrow, because they're given the opportunity for some reason, then they can present problems. And the first cluster on the test is carved out specifically under the banner of potential autoimmune triggers. And what we're going to look at a lot tonight are some of these organisms that commonly overgrow in the GI tract of our patients that are associated not only with higher incidences of various autoimmune conditions, but even now the genesis of those autoimmune conditions. We can look at additional dysbiotic or overgrowth bacteria that are not necessarily associated with autoimmunity, but can still be problematic. Also, fungi or yeasts such as Candida, Geotrichum, and others. And then we can look for other viruses associated with autoimmunity, such as cytomegalo and Epstein-Barr virus, and then various parasites in the protozoal class and even the helminth or worm class for identification. And finally, we'll look at a cluster of stool chemistries on the GI map that can tell us information about how we're digesting food, whether we're absorbing our food, what our immune response is like along the gut lining, do we have inflammation, are we at an elevated cancer risk, and do we have intestinal hyperpermeability, or what we call leaky gut syndrome. Now, having a tool like this is extremely important because we know the role of the gut microbiome is very important in the genesis of a whole host of systemic inflammatory diseases. This is clear in the medical literature right now. In fact, they are building signatures or classic aberrant patterns associated with a whole host of different inflammatory disorders that we can actually look for, screen for, as part of not only our diagnostic paradigm in diagnosing these conditions, but actually looking to pre-predict these conditions to find out who might be at risk for these conditions. And this would include various forms of colitis. This can include rheumatoid arthritis. This can include multiple sclerosis, and this can include many, many other autoimmune conditions. And this slide here just gives a rundown of just some of the microbes that can overgrow in the intestinal tract that are associated with various systemic pathologies. This is not an inclusive, exhaustive list. It's just a list until I ran out of room, quite frankly. And we're going to look at some of these associations, like Klebsiella and ankylosing spondylitis, Citrobacter, Klebsiella, Proteus, Prevotella, and rheumatoid arthritis. We're going to look at Fusobacterium and systemic sclerosis or scleroderma, Mycobacteria and psoriasis, Yersinia and autoimmune conditions of the thyroid, Streptococcus in pandas, and others. So to give you an example here, this is some emerging information just in late 2017 in the British Medical Journal, Gastroenterology. And this is the association between overgrowth of Fusobacterium and systemic sclerosis or scleroderma. That's just one example of these signatures that are being built for various disorders. But what I wanted to back up first is to talk just about a concept of why overgrowth of certain bacterium in the intestinal tract can actually help trigger an autoimmune attack against one of your host tissues or against a patient's host tissues. And what generally is occurring is these organisms on their surface have a bacteria or express some sort of peptide. This is usually a protein with a certain amino acid motif or sequence of amino acids that have a similar structure to the amino acid sequence on a receptor on, let's say, a host tissue, like the synovial lining of your joints, or the TSH receptors of your thyroid, or myelin proteins on your nervous system. And if there is a difference, but it's a subtle difference, a healthy immune system in a healthy individual can discriminate between the difference between those and does not confuse them. However, if you have a genetic susceptibility to overreact to one of these foreign proteins or peptides that are expressed on a bacterium, they may be associated with a virus, or they may be associated with a food protein that you're eating. And let's say you have a certain human leukocyte pattern or HLA pattern that makes you as an individual react to that peptide and hold it out to the rest of your immune cells and scream from the hills to your naive T cells, look at this structure. When you see this, know that this is not me. This is something that has to be dealt with and dealt with harshly. Basically, it's trying to teach your immune system this structure is an infection. It's an infective organism. And in the case of a bacteria overgrowing in the gut, that may very well be the case. However, if you have an immune system that is a little bit hyperactive, it's overcharged, you have a tendency to overreact, if you will, to that peptide, you can then lose the discrimination between that protein it learned from the bacterium and the protein that looks similar to it that is on your synovial joint linings or your thyroid or your nervous tissue. And if you start then not discriminating and crossing over and getting a reaction to that host tissue, if your reaction of your own immune system is to basically attack the synovial joints, it's rheumatoid arthritis or it's ankylosing spondylitis, depending on what anatomical joints are involved. If it's the nervous system, it could be MS or ALS. If it's the thyroid, it could be Hashimoto's or Graves' disease, and on and on. And this phenomena is simply known as molecular mimicry. One protein of a foreign source looks enough like a protein of one of your home tissues, if you will, where you no longer can discriminate between the two. And this is a wonderful paper that goes back a ways to 2008 called Joint Expedition, Linking Gut Inflammation to Arthritis. And this is a great paper to pull if you ever have time, that looks at how the dominoes fall from the overgrowth of a bacterium in the gut, such as Klebsiella and Citrobacter, to translation of these peptides through a leaky gut lining or a leaky mucosa, to overreaction by the host because of their HLA-B27 status, how that affects the systemic immune system to cross it over and essentially allow it to then attack the synovial linings of the joints. So this is an autoimmune arthritic model basically in rheumatoid arthritis. And if we back up even further, we see the initial emergence of some of these concepts. This is 1999 data from the Ukraine. Some of this stuff emerged first outside of the United States and outside of the research base of the United States. And you can see here, this group looked at the GI microbial composition of patients with rheumatoid arthritis versus those that didn't have rheumatoid arthritis. And what they found is things looked very different. The rheumatoid arthritis patients had overgrowth of bacteria such as Citrobacter, Klebsiella, and other enterobacteria that you did not see in normal people. But they went on further and they determined that when these patients with rheumatoid arthritis were in a very bad state, they were very symptomatic, they had a lot of problems going on with their disorder, their microbiota looked terrible in this pattern with Klebsiella and Citrobacter. But as autoimmune patients often do, they then go into periods where they feel a lot better and they're kind of in a quiescent state. When that happened in these rheumatoid arthritis patients, their GI microbiota looked like normal people. We see the same phenomena in ulcerative colitis, where it's really raging and they have 30, 40 bloody diarrheas a day and can't leave the house, in which case their microbiota looks like the aberrant pattern associated with that disease. But then when they all of a sudden resolve and they're in a really good state for a while, their microbiota looks normal. Which brings up the chicken or the egg question. Which shifts first? Does the disease flare, causing the GI microbiota to shift, or vice versa? And modern literature has effectively answered that question. And the answer is that the microbiota or the organisms overgrow and shift, which drives the disease into a symptomatic state. So we're learning more and more about a lot of these autoimmune diseases. And this is a paper with ankylosing spondylitis, ulcerative colitis, Crohn's disease, showing overgrowth of Klebsiella, of Proteus, and other types of organisms. So back to the GI map. In the autoimmune trigger situation, or section, I should say, you see this situation on a patient from my clinic, actually. This patient had been recently diagnosed with rheumatoid arthritis at an Ivy League Center of Excellence in Rheumatology, and they were basically offered methotrexate, oncotherapy, chemotherapy drug used off-label for rheumatoid arthritis, and a host of different highly suppressive immune agents known as biologics or response modifiers. Those medications can significantly help with symptoms, can significantly slow joint destruction, but unfortunately they come with the prospect of some significant side effects such as lymphoma, leukemia, and various forms of cancer, as well as opportunistic infections. So this person was looking for some other types of approaches, had heard about the functional medicine approach, and had come in to discuss this with us. And we talked about the various types of triggers, mediators, and antecedents that we looked for autoimmune diseases in functional medicine, and particularly in their case in rheumatoid arthritis. And we discussed looking at their GI microbiota with a GI map, and that we were looking for certain bacteria, including Citrobacter, Klebsiella, Proteus, Prevotella, and some others. And this person actually had elevated DNA recovery for 3 of those various organisms. And part of the treatment paradigm that we employ in these patients is to take these straws off the camel's back, if you will, to try to eradicate these organisms and reconstitute a healthier, less immunogenic microbiota composition in the gut. We have other organisms emerging in rheumatoid arthritis specifically, such as Prevotella. And if you look now at the expanded GI map targets for autoimmune triggers, you will see Proteus mirabilis, but also Prevotella just above that. But I did want to talk about Proteus a little bit more. Proteus as a species in general, but Proteus mirabilis, the species specific, the genus is Proteus, the species is mirabilis, is highly associated with rheumatoid arthritis. And some of this seminal work was done by a British physician by the name of Alan Embriger, Sir Alan Embriger. He's an MD, PhD researcher at King's College in London. He spent the better part of his career looking at alternate etiologies of rheumatoid arthritis, including bacterial etiologies of Proteus mirabilis stealth infections in the GI tract, but also in the urinary tract. Embriger's work has since been replicated all over the world, so this is not a phenomena unique to the English that we're going to talk about, nor is it unique to the English environment. It is something that can occur all over the globe. And basically what Embriger was able to prove is that some patients with RA develop RA, at least partly, because they harbor an ongoing stealth infection. By stealth, we mean it doesn't produce obvious side effects or obvious effects clinically to the patient, with an overgrowth of Proteus in their GI tract, but also particularly in their urinary tract. This is not the classic urinary tract infection that we learned in school, usually with E. coli that progresses to pain on urination and frequency and kidney infection and so forth. This bacterial infection can kind of lay dormant, kind of interacting with the immune system, but not causing overt symptoms. But the bacterium is producing metabolites, including a bacterial hemolysin and a urease enzyme, which the immune system reacts to. And this causes a cross-reaction with specific host tissues in the body, including the synovial joint lining, leading clinically to the disorder we would call rheumatoid arthritis. And what Embriger was able to do is actually elucidate the structures of similarity between the attacked host tissue, the attacked tissue in the patient, and the metabolites being produced by the Proteus organism. And he answered in the process many of the things we learned about diagnosing rheumatoid arthritis in school, whether it was medical school or chiropractic school or any other types of clinical training. We pretty much learned that rheumatoid arthritis has a strong female predominance, at least 3-to-1. Well, so do urinary tract infections. Women are much more prone to UTIs because of the differences in anatomy. Disease onset of RA between 30 and 50 years is very common. This is the exact age band in which women start being much more susceptible to chronic urinary tract infections. Exacerbation of RA after pregnancy. This is also seen with urinary tract infections, particularly after natural childbirth and more anatomical derangement, which can lead to higher likelihood of infections, particularly infections spread from the GI tract to the urinary tract. Low concordance rate in identical twins suggests that there's a non-genetic environmental factor, at least partly in play here, which a pathogen would be. Rheumatoid factors being high in RA can be a secondary phenomena due to the B cell stimulation because of the presence of the antigen-antibody complexes formed against Proteus. But finally, the real aha moment is that the hemolysin produced by the Proteus has an amino acid sequence or motif that very much looks like the HLA-DR molecules associated with the immune system of RA patients. And the Proteus urease enzyme that's produced and secreted by this organism in the urinary tract has a structural similarity to type 11 collagen. And type 11 collagen is basically only found in very few places in the body. And those places include all of the joints affected by rheumatoid arthritis, such as the small joints of the hand and wrist, and it is not found in the joints that are spared by rheumatoid arthritis. So in many ways, Embriger answered the riddle of rheumatoid arthritis with this model. And it's interesting that his model of treatment is something we would think is more akin to naturopathic medicine here in the United States, in that he does not use NSAIDs, DMARDs, immunosuppressants straight away. He actually uses antimicrobial therapy. In his case, he uses prescription antibiotics in the fluoroquinolone class, like Cipro, which we very much avoid because they're very tough on the mitochondria. But he uses herbal diets, he calls them, which include high levels of cranberry juice because of the D-mannose and blocking adhesion of these bacterium to the bladder wall, high fluid intake to diurese the patient, urine acidification with high-dose vitamin C. So this is the same kind of thing we would do in, let's say, a urinary tract infection by giving someone D-mannose powder, lots of organic cranberry juice, botanicals like hibiscus, aloe, and others. The other thing with autoimmune disorders in RA is to look beyond just one type or genus or species of microbe and look at the larger family of microbes, or what's called the phyla, the Bacteroidetes phyla or the Firmicutes phyla. These are large families of organisms, and we know when these ratios are not correct, it can cause immune imbalance. So we open up the lens very wide in the qPCR method on the GI map to also look at these things at the phyla level or at the large family level and do the ratios of those to determine if that might be creating an immune imbalance or if it might be contributing to other issues such as obesity. This is the so-called adiposity ratio because the Firmicutes organisms, if you have more of those and less Bacteroidetes, you will extract more calories from your food, including mainly your starches and carbohydrates. Crohn's disease is another autoimmune inflammatory bowel disease, which is now at least one variant of it is associated with mycobacterial infection in the GI, in the walls of the GI tract. Oftentimes, this mycobacterium is seeded from dairy products or contact with animals that harbor mycobacteria, such as cattle, such as birds. In fact, on the GI map, we have Mycobacterium avium because this is an organism strongly associated with Crohn's disease. Thyroid disease is another epidemic in our society. Hashimoto's and Graves', and particularly Hashimoto's, is growing at epidemic proportions. Molecular mimicry due to bacterial overgrowth in the gut is strongly implicated, particularly with Yersinia enterocolitica, which has peptides on its surface that look almost exactly like TSH receptors. So the body makes antibodies to bind to the antigen on the Yersinia organism to tag it that look like TSH receptors. They can mistakenly actually bind to TSH receptors, inadvertently directing an immune response against your own thyroid. And that happens very, very commonly. Although thyroid autoimmunity can also be triggered by various types of viral infections, including Epstein-Barr virus and cytomegalovirus. This is another reason why they are also represented on the GI map. Because while these stealth viruses can often be present in a stealth overgrowing manner in systemically in the blood, they are usually also concomitantly found in the GI tract. So you see here in this patient who came into my office, she was a young female. She had not been diagnosed with thyroid disorder, but she came in complaining of inability to lose weight, recently gaining weight, lack of exercise tolerance, generalized fatigue, IBS and constipation, and feeling cold all the time. And when we took her family history, her mother was on Synthroid, her older sister was on Synthroid, she had aunts on Synthroid, she had a very strong family history in older females in her family, or older than her, I should say, of autoimmune thyroiditis. And when we checked her GI map, she had Yersinia enterocolitica DNA at a much higher recovery rate than would be considered typical. And also, we look to see if she's having any reaction to gluten and gliadin containing fieldgrass grains, because, for instance, celiac patients, patients who have an extreme reaction to gluten and gliadin, have 10 times the rate of autoimmune thyroid diseases such as Graves' and Hashimoto's. So this is the same patient with the Yersinia enterocolitica positive, but in the additional tests in immunology, we see elevated total secretory IgA and elevated anti-gliadin secretory IgA. This suggests an intolerance to gluten and gliadin right along the gastric mucosa with mucosal immunology. So part of the treatment of this patient was to recommend to her a gluten-free diet, but also we use therapies to eradicate the Yersinia and to constitute a better microbiota. And now I'm going to show you some of the types of options you have to try to actually manipulate this microbiota clinically if you find these types of findings. So whether we're finding Klebsiella, Citrobacter, Yersinia, any of these opportunistic organisms, or even the pathogenic organisms that we do not want to be there, we often intervene with natural GI antimicrobial formulary products such as Microbe Essentials from Avexia Nutraceuticals. And you can see here there's a multitude of standardized botanicals in here, including multiple herbs or botanicals that contain the substance berberine. Berberine is highly antimicrobial to opportunistic organisms. We have berberine sulfate from Berberis aristata. We have barberry or Arctostaphylos uva ursi, standardized to arbutin. We have Juglans nigra or black walnut. We have barberry, also a berberine-containing herb. We have artemisinin or sweet wormwood, caprylic acid, and a wonderful antimicrobial, GI antimicrobial known as Tribulus terrestris. We often will combine this with oil of oregano therapy. Oil of oregano as a volatile oil is usually in a soft gel, where the kind of botanical extracts in Microbe Essentials is in a powdered capsule. So these are used in tandem with one another, usually 2 to 3 capsules of the Microbe Essentials with 1 or 2 of the oil of oregano, anywhere from 2 to 4 times a day, depending on the type of infection, the size of the individual, for anywhere from 2 to 4 weeks in between meals, while at the same time we're giving probiotics with meals. In the case of Helicobacter pylori overgrowth, like this case here, with positive virulence factors, meaning this is a potentially troublesome H. pylori, we often will use slightly different therapy, such as GastroBalance Plus from Avexia. This has a lot of anti-Helicobacter pylori substances, such as deglycerized licorice, Pistacia lentiscus, methylmethionine sulfonium, or so-called vitamin U. This is the factor in fresh cabbage, cabbage juice that is been known for many years as an anti-ulcerative, but also zinc carnosine, a highly anti-ulcerative, anti-H. pylori substance. We also look on the GI map at the normal commensal bacteria. So you can see on this example, for instance, we had very low bifidobacteria and very low lactobacillus. This is a case where you need to intervene with probiotic therapy. So something like Avexia's Probiotic Spheroids Plus gives a broad-based live culture probiotic that's delivered in specific, what are called probiospheres. They're not 2-piece capsules. These are specific delivery spheres that look a lot like soft gels, but they're constructed differently. They shield these organisms from moisture to keep them more viable on the shelf, in the bottle, and also while they're transiting your upper GI tract, so they can release where they're needed in the lower small intestine and the upper intestine, or I'm sorry, and the large intestine. Probiotic Essentials is just a larger dose of the same type of product. So the Probiotic Spheroids Plus is a 5 billion CFU, and the Probiotic Essentials is a 15 billion CFU. You do not really have to go much beyond these types of CFU counts when you're delivering the organisms in a spherate versus a capsule. And finally, probiotic microphage is a probiotic combination with what's called a bacteriophage. Phages are viral-like particles that are specific against specific types of bacteria. And the type of phage that is in probiotic microphage is anti-E. coli. So if you have overgrowth with E. coli, it can cause bowel symptoms, and it's associated with higher incidence of autoimmunity. This is where a probiotic blend with a phage can be very, very useful. Finally, in the pre and probiotic category, both, is probiotic myces. This is a Saccharomyces boulardii, or a specific brewer's yeast that promotes growth of beneficial flora. It attenuates growth of opportunistic and pathogenic bacteria, and it's particularly useful when there's any kind of overgrowth with Clostridia difficile, or C. diff. C. diff associated diarrhea, particularly after antibiotic use, is usually very well treated with Saccharomyces boulardii. In fact, in Europe, this is first-line drug therapy for antibiotic-associated diarrhea. Some of the digestive indicators on the GI map that are useful are things like pancreatic elastase 1 and steatocrit. Elastase basically tells you, are you producing enough pancreatic digestive enzymes? And steatocrit being too high means there's too much fat in the stool, so you're not digesting and absorbing fats. So I selected this sample because this patient would not be digesting their foods very well because they're not putting out enough pancreatic digestive enzymes, and they're leaving too much fat in the stool. They might benefit from a pancreatic digestive enzyme that's meant to digest proteins, like Digest Pro enzymes, or liver plus GB support. This is liver and gallbladder support. This thins the bile, keeps biliary flow going. In the case of trying to emulsify fats, absorb them, digest and absorb them better, the liver plus GB support is usually very effective at pulling down that steatocrit level if it's persistently elevated. Finally, I want to talk a little bit about the health of the GI mucosal lining, or the lining of the intestines. It's very, very important because it's a 1-2 punch. It really is important what kind of microbial balance you have in the gut, but also it's very important that you don't have hyperpermeability or leaky gut, or any kind of persistent damage going on to erode the gut lining, such as reactions to gliadin and gluten in your food. So we already looked at an example where we had elevated anti-gliadin secretory IgA. That's the case in this patient as well. Now, this may be because they're consuming lots of gluten, they're developing these antibodies, it's creating inflammation. We can see the inflammation because of the elevated calprotectin being high. That means there's inflammation going on in the gut. And then when we look down all the way on the bottom, we can see that they're overproducing zonulin. Zonulin is produced by the enterocytes. It can actually produce leaky gut, or hyperpermeability, or opening of the junctions between the enterocytes. So this is an example of someone we would be highly suspect that had inflammatory-based leaky gut, at least partially driven by the gluten in their diet that they are reacting to. One thing is good is they're producing decent amounts of pancreatic elastase 1, so their pancreas is putting out decent levels of enzymes. They do not have elevated fat in the stool. They do not have beta-glucuronidase elevated, which means they're not deconjugating a lot of their estrogens, and they don't have blood in the stool, which is good. Now, elevated zonulin, it was originally thought was more of a sort of a genetically determined thing, and it was one of the things that make people susceptible to autoimmune disease. But we are finding now that there's actually overgrowth of various bacterium that are causing overproduction of zonulin and the production of small intestinal barrier function disorders like leaky gut. So once again, overgrowth of E. coli and Salmonella, we know stimulates zonulin production, where giving bifidobacteria, lactobacillus, and some of the beneficial bacteria that we're used to can help lower zonulin output, can help treat leaky gut, and can make one much less susceptible to the effects of gluten. Let's say if you ate it and you had immune intolerance to it. So changes in the intestinal microbial composition can definitely directly cause leaky gut syndrome. This is a rather new thing in the literature. So trying to heal up the gut lining is extremely important as part of our intervention. Yeah, we want to kill the bad bacteria if we find it. We want to try to put in more of the good bacteria. We want to try to help digestion with digestive enzymes and all of the things we looked at, but we have to treat the leaky gut. This is a wonderful product called GastroPlus by Avexia. This is a powdered product that has L-glutamine, first of all, the preferred fuel source for the enterocytes to keep them healthy. But it has a lot of things in it that are immune proliferation blockers. These are glycobiology substances that are made of mucopolysaccharides, such as N-acetyl D-glucosamine, not glucosamine sulfate, similar to glucosamine sulfate, but actually different. But also a lot of what are called mucilaginous botanicals or herbs. These are your slippy, slimy stuff. Aloe vera, slippery elm, marshmallow, chamomile, okra, cat's claw. Think of some of these botanicals. Think of okra, for instance. If you ever cook okra and you get all that snotty stuff, that slimy stuff. Same thing with, think of the gel of the aloe vera plant. Slippery elm, it even tells you it's slippery, right? That's all the characteristics of these mucopolysaccharides, these sort of gel-like substances. They actually get in the gears between the antigen and the antibody, or between the antigen presenting cell and the naive T cell when it's presenting that environmental antigen that people often overreact to. These are immune blockers in the gut. We use them a lot medicinally in autoimmune diseases, both inflammatory bowel diseases, but also, for instance, there's a lot of trials going on in N-acetylglucosamine treatment in various chronic inflammatory bowel diseases, but also in multiple sclerosis, because it works in mechanisms very distinct from the conventional treatments or the drugs. And basically, the way these N-acetylglucosamine and these mucilaginous herbs work is by forming branches or lattices along the cell surface that prevents clustering of antigens. So this graphic really sums it up. If you have an antigen that your antigen presenting cell is trying to present to your T cell to activate it, it needs to fit like a lock and key. And when that happens, you get all of the signals that stimulate cell proliferation and immune reaction. But if you look at the graphic below, you have these mucopolysaccharide substances that kind of form a lattice through the lock and key mechanism. It's like sticking chewing gum in your padlock and then trying to get the key in. It just won't work. So that's some of the more modern natural therapeutics in stopping immune proliferation in autoimmunity. Now, some of these concepts that I talked about tonight are explored in detail in a couple of different papers of mine. One is in the Townsend letter for doctors and patients. The other is in the Open Journal of Rheumatology and Autoimmune Diseases. They're available full text on both of those publications, but probably the easiest way to get them is to simply visit my website, drdavidbrady.com, just drdavidbrady.com. In the media tab, under articles, you will be able to find the full text PDFs of those papers. And I wanted to end tonight with just two quick case studies showing the use of the GI map and the Avexia nutraceutical applications. This is a patient named Jason. Name has been changed, but he was a successful single guy, worked in the financial industry, very successful guy, very active, generally very healthy, ate out a lot, and he started having significant gastrointestinal distress. Went to a multitude of very high-profile, very top-notch gastroenterologists. They really couldn't find anything wrong with him. They did endoscopy. They did colonoscopy on multiple occasions. They did studies of his bile ducts. They did abdominal ultrasounds. They did conventional stool testing. They actually did some alternative stool testing using other methods, and they were not able to really determine what was causing any of his symptoms. So he had been worked up very comprehensively. And then when we GI mapped him, we found there was a little bit of DNA, but only at an equivocal level for Salmonella. We really don't think that that was clinically relevant, but we did find Cryptosporidium, a protozoal pathogen, in his gut, and we also found adenovirus. So he was harboring a virus in the intestines that can cause significant gastric distress, along with a protozoa. And he was an extensive traveler. He had traveled through the third world and the underdeveloped world a lot, and he was an adventurous eater when he was in those places. We don't know if that's where he picked up the Cryptosporidium or if he had had it for many, many years. When we looked further, his normal flora did not look bad. He had decent levels of lactobacillus and bifidobacteria, but in his immunology section, he was producing high levels of anti-gliadin secretory IgA, even in the face of low total secretory IgA. So if he was producing enough total secretory IgA to have what we would consider a very healthy, robust immune response at the mucosal level, he would have had very high anti-gliadin secretory IgA. So when we have anti-gliadin secretory IgA that's elevated, even when the total secretory IgA is low, we know that this person is very sensitive to gluten containing or gliadin containing grains. We intervened as follows. We put him on Gastromicrobe Essentials, this antimicrobial full spectrum product, 3 caps 3 times a day, or TID, away from meals. We put him on oil of oregano, 2 caps 3 times a day, away from meals. Both of those first 2 taken simultaneously. We put him on a liquid water clustered silver called silvercillin, mainly for the viral situation. We put him on a good probiotic, the probiotic microphage, 2 caps twice a day. This was with meals, so not taken at the same time as the antimicrobials. We put him on a gluten-free diet. We did all of this for the duration of 4 weeks. We let him have a week as a washout period. He was feeling clinically much, much better. In fact, virtually 100% resolved. And on retesting on GI mapping, there was no longer elevated Cryptosporidium DNA recovery or adenovirus. He still had the same pattern of slightly high Enterococcus. That's just his normal commensal flora pattern. We were able to get his total secretory IgA up a bit, but that seems to be also his sort of default pattern. But his anti-gliadin secretory IgA was now normalized. And then our second case was Nicole. Nicole came in with a host of different problems, including having had a diagnosis of irritable bowel syndrome. She didn't have any organic bowel diseases as worked up by a gastroenterologist, so no inflammatory bowel disease. She had been on many courses of antibiotics in her lifetime. And when we GI mapped her, we actually found Clostridia difficile toxin A genes. We also found Cryptosporidium and Entamoeba histolytica. She had also grown up in a situation where she essentially lived in a fairly rural environment and spent a lot of her childhood swimming in lakes, in streams, and so forth. We don't know if that's where she got the protozoal exposure or if that was more recent through food or travel, but it is what we found. She had low levels of lactobacillus or beneficial bacteria, but she also had elevated Klebsiella pneumoniae as an opportunistic organism, and this is related, of course, to autoimmune inflammatory arthritides, particularly rheumatoid arthritis, but also to some degree ankylosing spondylitis. Her treatment included Gastromicrobe Essentials, 2 caps 3 times a day away from meals. So we used only 2 instead of 3 caps because she was a much smaller individual than Jason was. 2 oil of oregano caps at the same time, actually soft gels. So the 2 first ingredients are the antimicrobials. They're taken 3 times a day away from meals, so on a relatively empty stomach if possible. She was put on a probiotic with meals, twice a day, probiotic microphage 2. She was also put on probiotic myces. This is the Saccharomyces product. This is because of her C. difficile, and she had been complaining of ongoing persistent diarrhea. And this is what had driven her to the gastroenterologist on a multitude of occasions. She had normal testing for stool analysis, and she did not come back with C. difficile. But remember, the standard testing done through normal major commercial labs is not quantitative PCR. It's not nearly as sensitive or specific. This was a 4-week duration protocol, on 1-week washout period, and on retesting, the protozoal DNA was clear, the C. difficile toxin DNA was clear, her probiotics were now within the normal range of lactobacillus, and the Klebsiella DNA was no longer present either. And with that, I will open it up to questions, and I'll turn it over to Michelle to talk a little bit about how you can order the Avexia nutraceuticals and the GI map, should you wish to.
All right. Thank you so much, Dr. Brady. Let's see. So the slide that we're on right now shows you the test menu and where you can actually order the GI map. You just log into your account, and if you don't have an account, obviously contact me and we'll set you up. Log into your account, go to the test menu, and click on the section for GI mapping. You can place the order right online, actually. And remember that it makes some sense to order a few kits at a time, because you're not charged for that kit until it's sent back to the lab. So, you know, if you have something on hand and somebody comes in and presents themselves with some of the issues that Dr. Brady has talked about, you would want to be able to have that available. And just by listening to those 2 case studies at the end, clearly you're going to want to run the test a second time once you put them on a regime of diet changes, nutraceuticals. Dr. Brady, before I get into our nutraceuticals, I do see a couple questions. Do you want me to read them to you, or are they available to you as well?
I can open up. Why don't you go ahead and read them so everyone knows what I'm
I can hear it.
Yeah.
Okay. So we have a question from Nicholas Hunley, if people don't mind. He says GI map test for blastocystis hominis.
Yep. Mm-hmm.
Yeah, that, and then question mark. I'm not sure if you can interpret that question for me a little.
If he's asking, do we test for blastocystis hominis? The answer is yes. The test is specific for that genus and species of protozoa. Yep.
Okay, perfect. So then we have another question. Okay. How did Jason and Nicole respond clinically after treatment? Yes, that's the question.
Good question. Jason actually did quite well for a prolonged amount of time. Actually, for over a year, he was pretty much symptom-free after we had optimized his gut from what we found on the initial GI map. He stayed off gluten-containing foods. It was actually only several weeks ago that he actually had contacted me and said he was having a minor relapse. He had been traveling a bit, was kind of eating some different things. And we actually have fairly recently ordered a new GI map, although we're still waiting on the results. It was just accessioned in the other day. In the interim, we did put him, after he collected his sample, on a sort of a very short course of that antimicrobial protocol that you saw on there, because he was just in some distress and he wanted some symptomatic relief. So we went ahead and sort of treated him like we did last time, and he actually started responding immediately. But I don't actually have the results back yet. Before we started treatment, to know exactly what was going on, but I look forward to seeing that. As far as the second patient goes, she's doing very well, and she was a little bit more complicated because her gut issues were only part of a much larger constellation of issues with her. We ended up having to really straighten out her thyroid physiology. She was pretty profoundly hypothyroid when she came into us, and we do think that that was a big part of her diagnosis of IBS, because when you're hypothyroid, you have very slow peristalsis and bowel function, and you tend to develop constipation, which throws your microbiota all off, and your whole gut ecology is not normal because of the slow transit time. So just getting her thyroid working again, I think, had a big part to do with helping her recover. She was also, we had organic acid tested her. She had some significant problems in mitochondrial ATP biochemistry, so we had addressed those. She was a little bit iron deficient, if I remember right. Her ferritin levels were down in the single digits, so we had to replete her iron. So we had to do a lot of things to kind of build her up. She came in as like a sort of a fibromyalgia, like, just really in bad shape kind of individual. So the gut stuff was the only thing I showed as part of her treatment, but we really treated her pretty comprehensively, and she's doing great long term.
That's great, Dr. Brady. I do want to mention to everyone that some of the tests that Dr. Brady just mentioned, the organic acid test, we have available. We have SIBO available. So I guess the point is that the GI map is where you start, but obviously if you need to do some follow-up, we have an Ask the Doctor feature that you can access right through your account. And if you want to ask about additional tests that might be needed, such as the organic acid test or SIBO, so make sure you utilize those tests from Avexia as well.
Yeah, I use organic acids all the time, Michelle. It's probably, besides, just normal standard serology, basic blood work, the two tests I probably use more than any other. I use a lot of them, but the two I use more than any other would be, GI mapping and organic acid testing.
Okay, great. So we have both of those available, and again, these are kits that you can keep in stock. So when people present with these conditions, you can test for them right away. We also have a question about H. pylori. If someone has a high H. pylori marker with negative virulence
factors. Yep.
Virulence, thank you. Would
Yep.
you recommend this to still be treated?
It's really a clinical call at that point. If the person has absolutely no symptoms, no GERD, no reflux, no gastritis, duodenitis symptoms, they've never had a positive endoscopy for ulceration. They have some elevated DNA recovery for H. pylori, but none of the virulence factors, then we usually don't treat that just as a matter of course. If the DNA recovery is exceedingly elevated, we might do so. I don't see many of those. If you're in doubt, I would suggest you call clinical support and talk to clinical support about the specific finding that you have and the clinical context that it's in, and then sort of make a decision from there.
Yeah, it looks like we have quite a couple of questions on the H. pylori. Let's see, I'm trying to get through all of them and not keep you all night, Dr. Brady. Let's see. What is standard on the test, and when would you choose the additional test? Then it goes that you test for virulence factors for H. pylori, and they talk about that again, the H. pylori. And it looks like people want a little more info on that.
Okay. Well, the standard test is the GI map, GI Microbial Assay Plus, and that basically has everything on it that I talked about tonight. There's one exception, and that's zonulin. the fecal zonulin marker for leaky gut is an add-on. So if you do the GI map and you want to add zonulin, you can see on the very bottom of that slide that's on the screen now, you'll see the add-on for zonulin, okay? So it's a fairly small, modest add-on charge. You can also order just a fecal or stool zonulin, but the cost is higher to do that, obviously, because the lab is only doing that one test. The GI pathogens profile is just a carve-out smaller panel of a section of the GI map. And it's the section of the GI map that was on page 1 that said pathogens. So if you, let's say you do a full GI map, and the only thing that comes back abnormal is you identify a pathogen or two that you need to treat. Well, with pathogens, you really need to make sure that you successfully eradicate them. So when you do the retest, if the only thing that was positive was pathogens, you don't need to do the entire GI map. You can simply do the GI pathogens profile. And the H. pylori profile, which is the H. pylori DNA and all the virulence factors, is also available as a carve-out separate test. But if you do the GI map, the full GI map, the one on the top, you are getting the GI pathogens profile, the H. pylori profile, all of the stool chemistries, all of the opportunist, all of the commensal or beneficial organisms, everything you saw that we went over tonight. The only decision point is, do you want zonulin or not? That's the add-on.
And I think, Dr. Brady, all they need to do is just check that off on the requisition that gets sent back.
Correct. The requisition form has all of these things that are listed on the slide, and it's a little box that you check, essentially.
Okay. And then I'll just end with this question about the nutraceuticals. And if there are other questions that come in, please, everyone, just go ahead and send them to me, to my email, michelle.holmes@avexiadiagnostics.com, and I will certainly follow up with Dr. Brady or Dr. Sedano on a response. But with respect to the Avexia nutraceuticals, why are you using certain preservatives in the products, and are they organic or how tested? We'll look more, and then they're going to actually look more into the products, which is great. So I guess they're just wanting to have some info on the ingredients.
Well, they're produced in a NSF GMP production facility. They're made of the highest, quality in the professional nutraceutical industry. Is every single component of every single ingredient of every single product organic? The answer is no, nor will it be in any nutraceutical line, because there are simply lots of different substances that are simply not commercially available organic. Whenever they are available organic, they are organic. What was the other question on there? It's used with the botanicals are all standardized. The vitamins are all in the preferred forms: phosphorylated B vitamins, albion chelated minerals. What was there another specific question,
I think that
Michelle?
was just about it. They were asking how the nutraceuticals were tested, but I think you spoke to that at the very beginning of your response.
Yeah, it's all NSF GMP, FDA inspected facility. Basically, all of the raw ingredients come with certificates analysis from the actual supplier of the raw, but then they are tested immediately once they're in-house before they're inventoried. They're tested again when they're taken out of inventory before they're mixed, and they're tested post-formulation or post-mixing, and then post-encapsulation or soft gelling, which is, pretty standard procedure for professional, high-end nutraceuticals. And the testing is done with, it depends on the substance. It could be near infrared spectrometry. It could be HPLC, quantitative analysis. There's a lot of different ways that you test depending on what the substance is you're trying to align to.
And what I will do tomorrow when I send this link, just so everyone knows, I see some of my clients that have participated tonight that have already been inquiring about the nutraceutical line. I've been sending out catalog information. Our website will be ready in 2 weeks, and our formal printed catalog will be mailed to all the clients. But in the meantime, remember that through March, we have the 5% discount on the Avexia nutraceuticals. Anyone can contact me directly, and I can help place an order. Obviously, looking back to, referring back to Dr. Brady's presentation for tonight, and I will have that information as well to help you guide which nutraceuticals to purchase for these conditions. Of course, they also come up in our functional health report if you decide you want to run some blood work. But all this information is going to be readily available in 2 weeks and is available through me right now. So I'm happy to help everybody. That's
Great.
it, Dr. Brady. Thank you so much for coming out tonight.
Yeah, I appreciate it. Thank you very much. Good night.
Good night, Dr. Brady.